Hi guys, it's Rob from Royal Balls. Hot on the heels of the disaster clutch, we have another clutch hatching, and this one hatched on day 54, which I think is pushing things a little bit too far, but fortunately all of these guys are perfectly okay. So given the response and all the comments on the disaster clutch video and some of the very thoughtful and thought-provoking suggestions in the comments section, I wanted to take a quick look at this clutch, but also to have a look in a little bit more detail at egg development. What is going on inside the eggs during the time that they develop, and what might be going wrong that could cause deformities. All too often I feel the hobby writes off these things as incubation issues without really stopping to think about what that actually means and what the processes involved could be. So I'm going to discuss that in this video. Okay guys, hot on the heels of the disaster clutch that you saw in the last video is actually clutch 12, and clutch 11 is still in the incubator and there are no pippers yet. This was not due to hatch until the 15th of December, and today is the 9th. These eggs have hatched 6 days early on day 54. Now if you remember, I had pushed up my incubator temperatures a little bit this year in order to shorten the incubation time. And the interesting thing is, it has not affected the sex ratios. You saw in the last clutch, which was deformed, that 2 out of 3 remaining babies were actually males. Now my incubator, when I checked this morning, I was actually surprised to see this clutch had already hatched. And I was also a little bit concerned because my incubator was running at 90.5 to 91 Fahrenheit, which is as close to 32 centigrade as makes no difference, and that is a little bit too high. That's the reason why these babies have hatched on day 54, and that potentially is also a reason why the last clutch was deformed, and we're going to talk about that a little bit when we look at this clutch. I hear many times on discussions in Facebook people say, is it a genetic deformity or is it an incubation issue? And incubation issues are always mentioned. For me, incubation issues and genetic deformities are one and the same thing. Any incubation issues exacerbates a potential genetic issue, and I'm going to talk about that with this clutch in relation to the last clutch. So first of all, let's have a look. Yes, Pepper. Yes, come on. You want to get involved as well, don't you? This is Pepper. She's very curious. Go on, you sit down there. So day 54, all the eggs have pipped and 2 of them are actually out. And something we've talked about before, there's one bad egg here. This died sometime during the incubation, and I did not notice because I don't really check my eggs in the incubator. I do a quick sniff test and there was no bad smell, but this egg is quite clearly dead, and it has not affected the other eggs. So proof positive right here in this clutch that a bad egg does not make the other eggs go bad. So we have 2 snakes already out, and we have this one bad egg, which is actually quite rubbery. It's gone very firm and dried out, which is the reason it doesn't smell. So let's just get rid of that. And no, I am not going to cut it to see what's inside. I think there are enough videos on YouTube showing that. Okay, so let's just zoom in on these remaining eggs here. And the first thing that you'll notice is that the eggs that have pipped by themselves, first of all, the eggshells have gone transparent where the fluid has spilled out on them, and you'll also notice that they're a little bit greenish coloured. Why do they get that greenish colour when they pip? Okay, well, inside the egg we've got the albumin, which is actually an antiseptic or bactericide fluid that prevents infection in the babies. And that is kept inside the egg by not one membrane, but actually there are several layers of membranes here. And each of those layers has a function. As your snake grows and develops, particularly over the last few weeks, it's going to be born with a fully developed digestive system and a fully developed urinary system. It has to, in order to survive when it comes out of the shell. Yes, Pepper? You want to be in the video? Yeah? So what happens in the last few weeks is that your baby snake has actually been peeing inside the shell, and one of those membrane envelopes, the function of that little pocket inside the egg is to keep the urinary products separate from the egg so it doesn't poison the egg. And when the snake cuts its way out, it punctures through all of those membranes, and those urinary fluids actually spill out onto the egg, which is why you get the slightly greenish colour. The urinary products from the egg are actually a slightly greenish tint. So when these eggs are pipped, not only are they now exposed to bacteria from the outside world, the contents of all those metabolic products have all spilled out onto the egg and also mixed with the albumin. So the clock is ticking. These babies cannot stay in the egg indefinitely. They have to get out because decomposition of the fluids within the egg has already started. So hopefully the babies have already absorbed all of that yolk sac and they're ready to come out of the egg, because if they don't, that fluid inside the egg is going to go bad. So for all those of you that cut eggs early before you see pippers that say you do not do any harm to the babies, you are wrong. You are penetrating that membrane and spilling out the contents of those urinary products into the egg. And if you cut early, the baby's going to have to sit in there for 2 or 3 days or even more before it comes out. So you are potentially doing the babies some harm by cutting early. You're not doing any harm if you cut at this stage. All these eggs have already pipped by themselves. They're all ready to come out. 2 of the babies here are already out all by themselves. And so if you wait for 1 or 2 pippers and then cut, you're probably okay. But if you cut early and there are no pippers, don't try to tell me that you're not doing the eggs any harm. You are cutting through those membranes, which all have a specific function related to the last couple of weeks of the snake's development inside the egg. And one of those functions is to hold the urinary products from the snake. It is actually excreting urinary products via the blood network inside this shell into that pocket inside the membranes, which are designed to hold those urinary products until the egg hatches. That's why the egg goes green. That's why if you cut early, very often the fluid inside the egg will start to decompose. It gets bacteria in it and it goes bad. The snake has to get out once it's cut through that membrane. The clock is ticking and that snake has to get out within a limited period of time. Now, because these eggs hatched so quickly, I have actually done a quick check on these guys just to make sure that they're not actually deformed and there is no shark jaw. And this one is okay, and this one is also okay. There do not appear to be any deformities in this clutch, despite the fact that they've hatched so quickly. And initially, these two babies here, you can see that they both have the classic leopard pattern. They have the dot on the head, there's the dot on the head stamp, and they have the extended, you can see they have the extended alien heads along the body. So a typical leopard pattern. So it's a pastel leopard paired to an orange dream yellow belly, and we can see immediately that these two snakes are different colours. This is much lighter than this one. This one does not have pastel. It also does not have orange dream. How do I know it doesn't have orange dream? Because the pattern is unaffected, and leopard orange dreams have a really wacky pattern. This one is very orange because it's actually yellow belly. You can see in person that the belly has actually got this kaleidoscope pattern on it. So this is a leopard yellow belly. This one, you can see the head stamp is very blushed, so this is obviously pastel. It is not yellow belly, and it is not orange dream. I don't see any pattern or colour enhancement over a standard pastel leopard. Again, you can see the leopard with the extended alien heads here, and pastel from the blushed out head stamp. So we've got one leopard yellow belly and one pastel leopard. We have not seen any orange dream, and we have 3 eggs left. So let's cut them and see what we got, and you will see hopefully the effect that orange dream has when it interacts with leopard, particularly if yellow belly is involved. Yellow belly tends to busy up the pattern. So let's cut these eggs now and see what's in the remaining 3. Hopefully we won't have a disaster like the last clutch, and all of these babies in here will be okay. So let's just cut this. It has already pipped. Okay, so this one is not leopard but is orange dream, I think, and it's probably a pastel orange dream. Also yellow belly, so maybe pastel yellow belly orange dream. You can see the yellow belly here on the flaming up the belly. Yes, the head is out, so now I can see that it is pastel. So I think this one is pastel yellow belly, possibly orange dream. Let's wait until it comes out. Let me just check this one also. I'm paranoid now, so let me check this one for shark jaw as well. And you can see that that one is perfectly okay. So this clutch does look okay. Let's get the next egg. Come on, I need some orange dream here. This egg is very full. It's quite difficult to cut here. Okay, so what do we have here? No leopard in this one either. Possibly just a yellow belly. I'm not seeing orange dream. So this one, I think just a yellow belly. Come on, head out. Thank you. So yes, the head is quite dark, so I'm saying yellow belly. And also this one is not deformed. There is no shark jaw in this one, thank goodness. So last egg, and I haven't seen any clear orange dream in these yet. Alright, this one's another leopard. It's a pastel leopard. You can see from the blushed head stamp, and you can see from the pattern quite clearly it's leopard, and I think this is just pastel leopard. So we'll have to wait for those to come out before we can try and identify Orange Dream, but Orange Dream is generally a very strong pattern modification, and I'm not seeing that in these snakes. So let's wait for them to come out. Okay, now that I know this clutch is okay, it's a little bit of thunder outside, it's about to have a tropical downpour outside. Let's talk a little bit about incubation issues versus genetic deformities. Now, we've talked before about the chromosomes and genes that are in these animals, and we tend to focus on the pattern and colour enhancing genes, but obviously the genetic code contains all the information needed to produce a snake. So what those genes do is control the production of hormones. Those hormones switch on and switch off the generation of certain types of proteins required for specialist cell development. So there will be, on the DNA code, there will be a gene that controls, for instance, the hormone that produces a digestive system, or eyes, or a lower jaw, for instance. And each of those genes has a very specific function. It requires the hormones to be switched on and off in concert with all the other genes on all the other chromosomes in order to produce a perfectly formed baby snake. Several things can go wrong with that. The gene itself could be damaged and may not produce the hormone, or may not produce the hormone in sufficient quantities to produce a perfectly developed snake. The receptors for those hormones may not be functioning, so the hormones may actually be produced, but the receptors are not responding, meaning that certain types of proteins in the development of the snake fail to be produced, and we get a deformed snake. So incubation versus genetic deformities are exactly the same. If something goes wrong, the gene doesn't work, the hormone receptors are maybe not functioning properly, and the correct proteins are not produced in the correct sequence in order to produce a non-deformed snake. And that can happen in the incubator. That can be a genetic deformity. Typically in our incubators we worry about temperature and humidity. If your humidity is wrong, your eggs are either going to go mouldy and die because they get too wet, or they're going to dehydrate and die because they're not wet enough. What generally happens during incubation is that temperature controls the speed of cell division. So the warmer the temperature, the faster cell division occurs, and that's why these babies have hatched in 54 days. Cell division has been so rapid that these babies are fully developed in 54 days. What that means is that if there is any potential genetic deformity inherited from the parents, or because your incubator temperatures are so warm that cell division is occurring so rapidly the hormones cannot keep up, then we could potentially get a deformed snake. Now, ball pythons are diploid animals. They get a full set of chromosomes from dad and a full set of chromosomes from mum, and typically if one side is damaged in any way, the other side can compensate. And so provided that your incubation temperatures are optimal, you will still get a non-deformed baby. And these snakes are extremely resilient, and that is the normal outcome, a non-deformed baby. However, if you are running your incubation temperatures on the warm side, like these guys here hatching in 54 days because I'm running my incubator at 32 centigrade, that is a little bit too warm for comfort. That means that cell division is proceeding so rapidly that if there are any deformities at all inherited from that genetic suite that these guys have got from both mum and dad, either the hormones can't keep pace with cell division, there's not enough hormone to produce enough of the specialized proteins, or the hormone receptors are not responding fast enough, and again, the specialized proteins, the specialized cells that go to produce a snake are not developed fast enough, and we end up with snakes with shark jaw or kinks or other deformities, and they're all caused by a gene not functioning properly, a hormone receptor not functioning properly, and the various specialized proteins not being developed in order to allow a perfectly developed snake to form. So I do not differentiate between an inherited genetic deformity and a deformity that's caused by perhaps incubation issues. Incubation issues are actually nothing more than a genetic deformity which is highlighted by our incubation process. So our incubation process can help or hinder with inherited genetic deformities. So putting everything down to incubation temperatures, I think, is completely wrong. If we understand the process, we know that what happens in our incubator and what happens with genetic deformities is actually exactly the same process. If your DNA inherited from both parents has an inherent defect, incubation temperatures are going to make that worse, and we will often write that off as being an incubation issue when actually it's not. It's an inherited genetic issue that is either covered up or made worse by our incubation process. Now remember also that cell division starts from the moment that fertilization occurs, which is actually around about 50 days before the eggs are laid. A snake will go through a pre-lay shed about 14 to 20 days after it's ovulated, and then 30 days or so after that the snake will lay eggs. So half of the incubation process, if you want to cite incubation issues for deformities in our snakes, half the incubation process has happened before the eggs are even in our incubator. So it's just as likely that something went wrong in the tub of your gravid female. For me, I don't differentiate, they're all the same. These are all genetic deficiencies. Genetic deficiencies that show up regardless, or genetic deficiencies that show up because our incubation temperatures are very warm, our snakes develop very fast, and hormone production can't keep pace. Now, we do know, for instance, that with super black pastels, super cinnamons and 8 balls, that it has been suggested that lower incubation temperatures lower the incidence of deformities. Why should that be the case? Well, lower incubation temperatures mean slower cell division, and if any of those genes are defective but are still actually functioning, they just fall short in their hormone production, slower incubation, slower cell division means the hormones stand a better chance of keeping pace. So protein production for all those specialized cells can still function efficiently, and we'll get a non-deformed snake at a lower temperature, whereas if we incubate at a higher temperature and our snakes develop faster, the hormones from those defective genes can't keep pace and we end up with a deformed snake. So yes, incubation temperatures do make a difference. Usually too high a temperature encourages deformities, but make no mistake, they are all inherent genetic deformities inherited from one parent or the other, which means that the argument of should I breed a deformed snake because it's only an incubation issue, it's not a genetic issue, is actually a non-argument. Deformed snakes have genes that did not function correctly, or hormone receptors that did not function correctly during the development of the babies, regardless of incubation. So you already know, if you have a deformed baby, that there's something wrong with some of the genes it contains, and therefore the likelihood of passing that on to its offspring, even if you think it's incubation issues, is likely to be increased. Incubation issues and genetic issues are actually one and the same thing. Alright, so this guy now has come out. This is a pastel. Woo! This is a pastel yellow belly. Does it have orange dream? The head is quite blushed, so maybe this is actually a pastel orange dream yellow belly. No leopard in this. Okay, so let's get these set up and we'll come back to these after they shed. But there you go, hot on the heels of a deformed clutch. I have a clutch that is not deformed but hatched in 54 days, which is too quick for comfort for me. And we've had a little bit of a chat about the difference between genetic deformities and incubation issues. And incubation issues are actually, for me, one and the same thing. These are all inherent genetic issues, either covered up or made worse by our incubation process. So there you go. I'm going to sex these as well, because we're right on the cusp of what I would consider to be close to lethal temperatures for these eggs. Any hotter and they would probably not develop at all, they would die. And of course we have talked in several videos about TSD, temperature sex determination. So I couldn't make these any warmer, and in temperature sex determination you reach a threshold temperature and all the babies should be one sex. If you don't reach the threshold temperature, all the babies should be the other sex. So in this particular case, I've heard it said that warmer temperatures produce more females. Well, if it's temperature sex determination, it shouldn't be more females, it should be all females. But let's have a look. These have been produced at very warm temperatures, and that actually is a female. This guy here, just out of the egg. Whoop. Now you'll see when I press gently that this baby, although it's straight from the egg, is already peeing. So that's what I was talking about in terms of the membranes that actually hold this pee until the snake comes out. Now that, despite the fact that this has been incubated at really high temperatures, is a male. That should not happen in temperature sex determination. This is genotypic sex determination. The sex was determined at fertilization. Let's just have a quick look at this one. That's actually a female. And we've got two in the egg that are not out yet. Alright. So all the babies are out now. Thankfully they are all perfectly okay, and we have 1, 2, 3 leopards and 2 which are not leopards. I was hoping to see in these babies an aberrant pattern caused by the interaction of leopard and OD. Let me show you what I mean. This guy here is a leopard orange dream yellow belly, and you can see what orange dream does to the pattern on this guy. Very, very distinctive. There is a ringer in the tail. We have these awesome train tracks from orange dream, which in this case are lifted quite high up the body. But you will notice that none of these guys have that pattern, so we do not have any leopard orange dreams. I'm going to save the identification until these have actually shed out, and we'll revisit these and take another look. But for the time being, just to reassure both you and me that these are all okay, I thought I'd just show you them all together. Now I have sexed them and I have actually already ID'd them, so I know what they are, but I'm going to save that until later. That's a female. That's also a female. Also a female. That's a male. And that's a male. So the sex ratios here are 3:2. Despite these hatching on day 54, we still have the sex ratios we would expect. It's within a smidgen of 50-50. There are only 5 snakes, so one of the sexes had to be 3, and in this case it's the female. But as we've talked about with TSD, there shouldn't be any mixed sexes. The sexes should all be one or the other if the incubation temperatures are actually affecting the sexes. And we can see that we got 2 males, even though these hatched on day 54, which is incredibly fast. It didn't stop these 2 males being males. Okay guys, thanks for watching. I hope you enjoyed looking at that clutch. It's a big relief to me, and we have another clutch has already hatched and more eggs have been laid, so it's actually getting quite hard to keep up with the videos. A lot more to come, and I want to continue this discussion about embryo development and things that might go wrong, and some more genetic discussion in part 2 of I Didn't Know That. So plenty more to come. I should have videos out every day if I can manage it, because I've already shot the videos and I just need to do the intros and the outros. So thanks for watching. Don't forget to share, like and subscribe. Stay tuned, and we'll see you for the next one.